Lateral gene transfer is the movement of genetic material between unicellular and multicellular organisms. In other words, the transmission of DNA from parent to offspring. It's an essential factor in the evolution of organisms. It's also known as horizontal gene transfer. It is the primary mechanism for the spread of antibiotic resistance or any other characteristic or traits between bacteria, and it played an important role in the evolution of bacteria and the evolution maintenance of transmission of virulence. Lateral gene transfer is the transmission of genes sometimes across species barriers with classic vertical inheritance from parent offspring. Lateral gene transfer is recognized as an important phenomenon that has shaped the genomes and biology of prokaryotes. As I move to my next slide, LGBT2 occurs with one adult bacterium transferring its genes to another either through conjugation, transformation, or transduction. In this tree, we see connection between bacteria, archaea, and eukaryotes. We have a common ancestor all the way at the bottom. And over time, over the years, over the decades, over the centuries, maybe millions and millions of years, we're all related to one common ancestor. In the third slide, there's one example I gave in the beginning about drug resistance. Horizontal gene transfer is common among bacteria, even among very distantly related ones. For example, drug resistance is an important topic as some bacteria have developed some sort of resistance towards drugs and pass it on to other species like E. coli or Salmonella. This is why over the years you are required a flu shot as the pathogen has evolved and developed a new drug resistance. So that explains why over the years you're recommended a flu shot, because the pathogen has gone stronger and developed an immunity towards it. As I said, that there's three ways of lateral gene transfer: conjugation, transformation, and transduction. Conjugation is a direct transfer of genes between bacteria through conjugation tubes. As you see in this figure, at A you can see two cells and one donor cell donating or passing on a DNA strand to the new recipient cell, causing a complete change in genetic variation in the DNA of the recipient cell. As we see, transformation is the genetic alteration of a cell resulting from direct uptake and incorporation. So here we see DNA fragments incorporating itself into the new DNA, and the recipient cell changes its DNA completely. Transduction is the process by which foreign DNA is introduced by a cell by a virus or viral vector. One example of this can come via parasite infect humans with a trypanosomal Kangas disease, which can insert DNA into the human genome. It has been suggested that lateral gene transfer to humans from bacteria may play a role in cancer. So this can also be a transduction can also be an parasitic relationship between the host and the parasite. In my final slide, so here's an example. As I said, parasite infecting humans. The parasite leads to a strand of its own DNA, which changes the DNA within humans, which can cause horrific diseases. In my last slide, I will talk about speciation and species. Species are separated from one another by prezygotic or post-zygotic barriers, which prevent mating or production of viable offspring. Speciation is a process in which new species form. It occurs when a group in a species become reproductively isolated and diverge. One example is the Galapagos finches. As we can see, there's a variety of species, but they all come from the same family. As we could see, they each have similar skull structures, but you can see there's a difference between beaks. This happened because each Galapagos finch was located in different areas of an island in the Galapagos, and each beak was adapted to eat seeds, as some beaks were made to crack open harder seeds, some were made to eat smaller seeds, bigger seeds. It all depends on where they were located. So that's my presentation over species and speciation and lateral gene transfer. Thank you.